The diagram shows the representation of desalination of water. This is a schematic representation where we have two compartments separated by a semi-permeable membrane. So the semi-permeable membrane used here is cellulose acetate, which is smeared over a support. Seawater contains less amount of solvent compared to that of fresh water. Now, when it is separated by a semi-permeable membrane and if we are applying an external pressure P, which is greater than that of the osmotic pressure, we find that reverse osmosis occurs. That is, movement of solvent molecules from the region of lower concentration to the region of high concentration occurs. That means water from seawater is forced to move across the semi-permeable membrane into fresh water, leading to desalination of water. So this is how the Gulf countries prepare drinking water from seawater. The process is known as desalination of water. A similar process of desalination is used in water filters, which is marked as RO. RO filtered water means it is reverse osmosis through which the filtration occurs. Nowadays, the water filters are using reverse osmosis, ozone gas, as well as activated charcoal for purifying water. You find in the process reverse osmosis occurs, then the germicidal property of ozone is shown. In addition to that, adsorption of particulate matter from dirty water also occurs, which ensures pure, clean drinking water. We are going to discuss what happens to the blood cell when placed in different concentration of sodium chloride. You know that blood cell is having same concentration as 0.91% sodium chloride. So you find that blood cell is isotonic with 0.91% sodium chloride. So whenever we place a blood cell in 0.91% sodium chloride solution, there will not be the change in the size of the cell because no osmosis occurs here. Whereas if you are placing the blood cell in a concentration higher than 0.91%, you find that concentration of the solvent water is more inside. As a result, what happens here? Concentration is more, and so we know that osmosis is a process of transfer or movement of molecules from a region of higher concentration to a region of lower concentration. As a result, it undergoes exosmosis. That means you find that the size of the cell will shrink in the case of the solution which contains less than 0.91% sodium chloride. When blood cell is placed, you find that concentration of the solvent will be more outside. As a result, what happens is water gushes inside, and as a result you find that the size of the cell will bulge. The cell will bulge in shape. So you find that when concentration is changed from 0.91%, either the cell will undergo shrinkage or the cell will burst as a result of bulging of the cell. So to avoid this, you find that always intravenous injections are taken by mixing it or diluting it with 0.91% sodium chloride so that there is no bulging or shrinkage of the cells.